Thank you so much. It's a great pleasure to be with you all virtually to discuss in silico experimentation from your living room. And as a breast medical oncologist and computational biologist who works at the intersection of both clinical oncology and big data, I'm hopeful that the session today will provide you with more tools to access and analyze existing data sets across the spectrum of breast cancer research. These are my disclosures. So today we're going to start by discussing principles and pitfalls of in silico experimentation because there's never a free lunch. Then we'll walk through some publicly available resources, both basic as well as clinical. And then we'll do an in silico group experiment to put these into play. So first, thinking about principles and pitfalls of in silico experimentation. When many people reference big data in the context of breast cancer, they think of bulk DNA or RNA sequencing. But clearly, big data is not just genomics. And even within genomics, the scope of large complex data sets is expanding. Single cell sequencing, ChIP-seq, ATAC-seq, spatial sequencing, cell-free sequencing. Proteomics is a field in and of itself. And there's a lot of other different types of big data, such as screens, digital pathology, and radiomics. And even clinical data these days can certainly be considered big data when we think about large repositories such as SEER or NCDB, as well as claims data or CancerLink or other real-world repositories. In defining the key ways that we as a community analyze and utilize big data, it's critical that we all have a shared set of principles to ensure access and usability of all of these data. FAIR principles have really been critical as a field and across fields to ensure that all publicly available data adheres to findability, accessibility, interoperability, and reuse of digital assets or ability to reuse data. Reproducibility is a major challenge across biomedical science, but particularly, I think, in computational analyses and bioinformatics. I love this review that was published in PLOS Comp Bio in 2013 that lays out 10 simple rules for reproducible computational research and a really straightforward 10-point checklist that we can all follow to ensure the analyses that we're doing are rigorous and reproducible. Speaking of rigor, analyses of computational data should be rigorous, just like any in vitro experiment or clinical trials. Or a clinical trial. Specifically, we should test hypotheses rather than sifting through data to look for statistically significant associations that we can put in figure 5B. We should outline a hypothesis and use the same rigor that is used in any other experimentation. This review in Giga Science in 2020 lays out how rigor needs to start all the way back at computational training, all the way through funding, and that we can encourage reproduction of results, both in our own analyses and in the field. Another key element of rigor is statistical rigor. As the complexity of data has increased, the appropriate use of the right statistical test has become more challenging. In addition, if we are running multiple tests on a single data set, we also have to account for multiple test correction, which can certainly be missed in some analyses. This, I think, reveals the importance of collaboration with biostatisticians to ensure that the analyses that we're doing are appropriate and statistically sound. One example that I see frequently is analyses of differential gene expression. While in many settings, the t-test is an appropriate statistical test to evaluate two continuous variables, it is clear over time that better or more appropriate computational and statistical approaches have been developed to analyze RNA sequencing data. And several of these have been benchmarked against one another, two of those papers that I've laid out here. So I think that this reinforces that not only do we need to test hypotheses, but we also need to ensure that the statistical analyses we're doing are rigorous and fit the data. We also need to address confounders. One example prominent in breast cancer is proliferation. When we think about gene expression in breast cancer cell lines, for example, comparison of a proliferating cell state versus a non-proliferating cell state is associated with changes in over 4,000 transcripts. We also know clinically that breast cancer prognosis is associated with grade, a marker of proliferation, among other things, as well as receptor or molecular subtype, each of which have their own association with proliferation, which is associated with proliferation gene expression. So we can see that this interrelationship has the potential to confound a variety of analyses. I think that this was brought very well into focus over the last decade, particularly in a couple of papers, one by David Vinay and another by Benjamin Habey-Keynes, where they showed that either random gene expression signatures, often those that correlate with proliferation, or a single proliferation gene can be associated with breast cancer outcomes, thus reinforcing the importance of understanding what the confounders are and accounting for them. Finally, today I'm going to touch on the inherent limitations of data. The Cancer Genome Atlas, or TCGA, is an incredible repository of multi-omic data, but it was really developed just for that, as a repository of multi-omics, not necessarily clinical outcomes. As laid out in the clinical data resource of TCGA, published as part of the PanCan analyses in Cell in 2018, TCGA breast cancer clinical data has some limitations. For example, when we look at overall survival and compare estrogen positive in light blue versus estrogen negative breast cancer in red, we can see that ER positive versus ER negative has no significant overall survival. We know within the field that this is not true. This is that these two receptor subtypes have different outcomes, and this has been demonstrated for decades. But this is more a limitation of the TCGA clinical data, which doesn't have the length of follow-up necessary to allow appropriate overall survival analyses. Disease-specific survival is more appropriate, but also has some limitations. And so I think that this leads to the importance of understanding the limitations of data, even large publicly available data sets like TCGA. So now that we have some idea of some principles and potential pitfalls, what about publicly available resources? There's an incredible amount out there on breast cancer cell lines. One of the most comprehensive is the Cancer Dependency Map, which subsumed the Cancer Cell Line Encyclopedia from the Broad Institute, where hundreds of cell lines are profiled omically, including RNA expression, DNA sequencing, as well as CRISPR or RNAi screens, as well as drug screens. And all this is publicly available and easily searchable. Steve Ethier from Medical University of South Carolina has an incredible some breast cancer cell line knowledge base that allows investigation of the some breast cancer cell lines that he was instrumental in developing. Beyond cell lines, we also have a growing set of data about mouse or patient-derived PDX models. The Jackson Labs has the Mouse Genome Informatics page, which allows the ability to interrogate common breast cancer mouse models. And PDX Network, which has a growing repository of breast cancer patient-derived xenografts, particularly from Baylor and Utah, has omics data coming soon that will allow greater understanding of the feature of the models that we use experimentally. When we think about clinical breast cancer data, one of the easiest and most widely utilized repositories is CBioPortal. CBioPortal, when you look at the breast cancer data sets, has a variety of breast cancer data sets in various settings, including primary, metastatic, and a variety of omic data. Looking at one of these, Metabrick, I think is important to highlight because it's a very important, long-standing, and powerful data set that has over 1,900 primary breast cancers, and of which there's gene expression, copy number, mutation data for 171 genes, as well as data such as long-term follow-up, site of metastasis, and even image mass cytometry. The Cancer Genome Atlas, which we've also mentioned, also has over 1,000 primary breast cancers, has gene expression and microRNA data, as well as copy number, more comprehensive mutation data, including whole exome sequencing, WES, and a subset that has whole genome sequencing, as well as other resources such as methylation and protein data. If one is interested to look at the process data, CBioPortal is an outstanding resource. But if you were more on the informatics side and wanted to obtain the raw sequencing data for subsequent analyses, you would gain controlled access and then be able to leverage the incredible resources with the Genomic Data Commons through the NCI Cancer Research Data Commons. The Cancer Research Data Commons has many different sort of nodes, including proteomics, the Genomic Data Commons being one of those, imaging, canine, and clinical trials. But it has a very remarkable repository of different data types. And we can see that breast cancer is among the most common. If we look at the exploration tab, we can see that we can go into a specific primary site like breast and even refine the characteristics of the samples that we're interested in. We can determine the number of sequencing files that are available for potential download. And we can even make an oncogrid or oncoplot of our samples of interest, looking at frequent mutations and copy number analyses with certain clinical tracks as well. I love the fact that they've also incorporated the digital pathology here, where one can just click on the slides segment and actually be able to view the H&E slide of that particular sample. The future, I think, is the acceleration of multi-omic data deposition. As part of the Alliance for Clinical Trials in Oncology, one of the NCI cooperative groups, we've been working to build our standardized translational omics resource, or ASTOR, which is a living repository of high-dimensional data from Alliance clinical trials, including rapid deposition of sequencing files, standardized pipelines to harmonize computational processing, co-localization with other data types like digital pathology or published clinical data, and some basic visualization software for non-informaticians. And with an outstanding group that's been working hard, our goal is to accelerate translational research from the years that it often takes from a report of clinical trials to potentially faster, even months of deposition of genomic data. So now that we've gone over principles and pitfalls, as well as some publicly available resources, I thought for this education session, it would be great to do an in silico group experiment to put these into practice. So let's experiment. If we're going to do an experiment, we need a hypothesis. And so for this exercise, I put together the hypothesis that genes included in clinical genomic predictors for hormone receptor positive, HER2 negative breast cancer may be potential targets, something that we could explore more. Our goal here is to generate preliminary data necessary for evaluating new targets in breast cancer. So acknowledging that this is a hypothesis generating experiment. We want to put together an analysis plan. And so for our gene of interest, we're going to explore available data sets to evaluate the association with outcome in patients to make sure it's relevant, identify appropriate cell line models that might be experimentally analyzed, identify putative drug targets, and then assess utility for further study. So first, let's pick our gene of interest. There was a recent review that profiled a variety of clinical genomic assays, such as Oncotype DX, MammaPrint, ProSigna, EndoPredict, BCI, MammaStrat, and IHC4 to look for overlapping genes across these various assays. There are some like BRC5 that are in multiple assays and are known to be associated with proliferation, which we mentioned previously. But I thought for the sake of this experiment, we'd focus on one GRB7. So what is GRB7? Well, we can turn to a great online gene data repository, gene cards, to look at the gene name, aliases, its location, and get a gene summary. In this case, the product of this gene belongs to a small family of adapter proteins known to interact with a number of receptor tyrosine kinases. Looking like a promising target so far. It's in genomic predictors. It interacts with receptor tyrosine kinases. Let's keep going. Next question is, does it matter in breast cancer? Well, we can turn to see Bioportal, click on breast cancer, go to Metabrick, and query by gene to then look at the RNA gene expression data, since these are primarily gene expression predictors that we were looking at, input our gene, and submit our query. With just those 6 clicks, we can identify that GRB7 is overexpressed in about 17% of breast cancers in the Metabrick cohorts. So a pretty decent number, not a rare phenomenon. We can add tracks to then increase our understanding of these patients by adding, for example, PAM50 profiling in tumor stage to see if there are any patterns associated with GRB overexpression. We can also move on to the survival assessment to see if this is relevant within breast cancer. And looking at relapse-free survival of over 1,900 patients, we can see that those patients who have overexpression of GRB7, the altered group in red, have significantly worse relapse-free survival relative to those who do not have overexpression of GRB7 in blue. And this is highly statistically significant, suggesting that this observation is likely relevant in breast cancer. If we go back to our query, we can also look to see what other things are associated with GRB7 overexpression. By looking at RNA expression on the y-axis and copy number alteration on the x-axis in the plots function, we can see that the majority of tumors that have overexpression of GRB7 actually have GRB7 amplification. So not only is it frequently overexpressed, it's frequently amplified, and the amplification is associated with high expression. Now, this is looking in one data set. We know that we have at our fingertips other data sets. So why don't we turn to an orthogonal data set, modify our query, and move over to the Cancer Genome Atlas. In TCGA, we can again see that those patients who have overexpression of GRB7 also have frequent amplifications. So we've sort of proven to ourselves in two data sets that, we're looking at high GRB expression and amplification. Can we identify cell lines with this genotypic characteristics for further investigation? We'll turn to the cancer dependency map. We'll put in GRB7 as our gene of interest. And this pulls up sort of an overview profile, including its enriched lineages, the structure, and so on. But we'll want to go on to Data Explorer to try and identify the specific cell lines that match our genotype of interest. In this Explorer, we can put the expression along the x-axis and the copy number along the y-axis by just inputting our gene into the search bars. And we can pretty quickly identify that there are a decent number of cell lines that have high copy number and high expression of GRB7, which matches what we saw in the patient data. These are primarily gastric, esophageal, and a variety of breast cancer cell lines, several of which I've labeled here. We can also look to see whether there's any available drug data to suggest that certain lines are associated with sensitivity with when GRB7 is highly expressed. By clicking on Show Precomputed Associations and looking at drug sensitivity, we can see that the top named hit is actually lapatinib, a small molecule inhibitor of HER2 and other kinases that's FDA approved for use in HER2 positive breast cancer. So this is a compelling hit, and since we're investigating breast cancer outcomes, may be of great interest. We can refine our search to actually look at GRB expression on the x-axis again, with lapatinib sensitivity on the y-axis from this particular drug sensitivity set of experiments. And those tumors that have high GRB7 expression seems to be more sensitive to lapatinib, as that initial query might suggest. But we know that there's multiple HER2 small molecule inhibitors, and actually we can query multiple of these as an orthogonal sort of check on what we're seeing. For example, we can look at neratinib, which also shows a similar trend, as does tucatinib. So I think we're starting to get a consistent set of features associated with our GRB7 overexpression as we're getting through this in silico experiment. One of those consistent associations is something to do with HER2. We see that GRB7 overexpression and amplification is primarily in HER2 positive cell lines, like HCC 1954 and BT474, among others. Further, HER2 small molecule inhibitors like lapatinib, neratinib, and tucatinib all seem to be active in GRB7 overexpressing lines. So let's go back to cBioportal and look at HER2 positive status and its association with GRB7. And in fact, what we find is GRB7 and ERBB2 or HER2 are almost always co-amplified. So if one is amplified, the other is amplified as well. So how could this happen? Well, When we look at gene cards, we can see that GRB7 and ERB2 are both located on 17q12. How far apart are they? We can actually turn to the UCSC genome browser and just on its main gateway page, type in GRB7 and find that ERB2 or HER2 and GRB7 are very close together, only a couple kilobases away on 17q12. So this suggests that these genes are actually so close together that when HER2 is amplified, GRB7 is also amplified or co-amplified at the same time. So this brings us back to our question about all breast cancers. We did this survival analysis suggesting that GRB7 overexpression was associated with poor survival. What about when we look within individual breast cancer subtypes? We actually find that none of these survival associations are significant. Now, how could that be? Well, in fact, GRB7 is a biomarker for HER2 positive breast cancer. And when we were looking across all breast cancer subtypes, we were actually just comparing HER2 positive breast cancers with non-HER2 positive breast cancers. So hopefully this example has shown us both the power of a lot of these publicly available tools and how pointing and clicking from our living room, we can learn a lot of data, but also some of the pitfalls. For example, that pan breast cancer analyses are a common cause of confusion if we don't do within subsets and don't take advantage of the fact that we know that there are existing subtypes within breast cancer. So I think this reinforces the importance of never going alone. As I say in cBioportal, interpret all results with caution as they can be confounded by many different variables. So most publicly available data, I think, is best used for hypothesis generation. And while rigorous secondary analyses or true experiments can be done using public data, it requires partnership with your friendly bioinformatician or computational biologist in collaboration with a biostatistician. I would reinforce that high quality, careful, rigorous analyses take time and expertise, so value your data analytic collaborators. View this as a collaboration, not a service, and include your bioinformaticians and statisticians as co-authors on papers that leverage these data, as co-Is on funding, and give them time to do these analyses, because doing it well often takes time. As I tell many of my collaborators, be nice to your biostatistician and bioinformatician, because if you need that revision figure on a tight timeline, where will you be when you need help? So in conclusion, we've gone over principles and pitfalls of in silico experimentation. We talked about resources for both bench or preclinical settings as well as clinical. We went through an in silico experiment, starting from a hypothesis and a plan through evaluation of clinical data, cell lines, drug activity, and genome browsing as well, with the importance of team science above all else. With that, I'd like to thank so much everyone in my lab, including the alumni, my colleagues and partners at Ohio State Breast Medical Oncology, my mentors, in particular, Laura Selfers, a bioinformatician in Joan Brugge's lab at Harvard, where I did my postdoc, who really started me on this pathway. And finally, my funding and of course, patients, advocates, my patients and patients who donated samples for these analyses. Thank you so much.